The overall goal of this procedure is to obtain the uniaxial high strain rate true stress strain response of the soft biomaterials being tested. This is accomplished by first testing a brain sample in a Split-Hopkinson Pressure Bar, or SHPB, apparatus, and then calibrating the one-dimensional version of the material model with experimental true stress-strain response. The finite element, or FE, simulation of the SHPB setup is then performed to evaluate the sample's average loading direction stress and centerline stress. The final step is to iteratively compare the one-dimensional material model fit and FE simulation centerline stress and the three-dimensional FE loading direction stress and experimental true stress-strain response until good agreement between the aforementioned results are obtained. Ultimately, the coupled experimental finite element methodology is used to show that the uniaxial true stress-strain response for soft biomaterials can be obtained using an SHPB.
The main advantage of this technique over existing methods like shock tube test is that this technique renders the high strain rate uniaxial mechanical response of a soft biomaterial. The implications of this technique extend towards the diagnosis of traumatic brain injury through virtual human body modeling, because the data obtained from the current method can accurately capture local injuries and damage arising from trauma to the human head.
We first had the idea for this method when working on correlating the brain high strain rate experiment data with the results from finite element modeling.
Demonstrating this procedure will be veterinarian Dr. Jim Cooley, graduate student Mr. Sourav.